Okay, sorry, technology is limited and it has a half an hour cut off. So I'm back. Anyway, describe how scientists could find out if badger vaccination reduces TB in cattle. Okay, so it's a describe question, so we don't have to think too hard. All right. If you're going to essentially, you know what this is? This is a design and investigation question. That is what this is. Okay. But just not in an experiment, that in a lab, like in a controlled setting, right? So it's kind of like Corms, okay? So, well, it is actually Corms, to be honest, but it's only 3 marks. So, what are we changing here? Okay, is if they have the vaccination or not. So we can say vaccinate one group of badgers or badgers in one area, badgers in one area, and not in another, or not another group. Okay, and then we would what we so that's our change. Okay, we'll come back to the organism in a second because obviously they should all be the same species of badger. We're not going to dispute that. We're not even going to state the obvious there, I mean. Let's think about the measure first of all. What are we measuring? I feel like there should be two M's, not two R's. There we go. Let's correct that. What are we measuring? We are measuring, so we want to see if it reduces TB in cattle, right? So measure or count. Let's say count. Count the number. Oh, should I write it out for you guys? The number of cattle that have TB. Okay, so obviously the fewer cattle means that, oh, hey, vaccination worked. Okay, so that's my measure. Now, I would say after a certain amount of time, but it's only 3 marks, so I'm going to disregard this M and I'm going to disregard the repeat. So let's start with the same. There's quite a few same things we can say here, but we only need one more mark. Same number of badges. Same. Sorry, maybe we should clarify. Test on the same number of badges and done, 3 marks. But we could also say same number of cattle, right? Because imagine you've only vaccinated 5 badgers and you're testing 5 badgers in one area, 5 in another, but one area has like 20 cattle and the other area has 2 cattle. So it's not going to be very helpful. So same number of badgers and also same number of cattle. Not cattles, sorry, cattle. And same number of cattle. Boom, done. Okay, 3 marks, lots of our columns there, and it's just our 3 variables, our independent, dependent, and control variables. Beautiful, wonderful. Right, now, selective breeding in snapdragons with brightly colored flowers. This one, you should know this one, I shouldn't even have to explain this. The answer follows the same pattern every single time. We want brightly colored flowers, that's our desired characteristic here, so select 2 plants with brightly colored flowers, bright flowers, can't be bothered to write the whole thing, bright flowers. Okay, and then what do we do with them? And breed them. Okay, so that's your pollen and egg cell. Okay, so we breed them and then we select the offspring with bright flowers and breed them as well. If it was 3 marks, I would say repeat over several generations with bright flowers and breed them together. Or cross pollinate, breed, whatever you want to say, cross them. Okay, done. So we've got offspring, we mentioned the characteristic, and we have mentioned breeding. Okay, nice and easy. Right, I believe this question is about micropropagation. Now we will do a lesson on cloning in plants because we talked about cloning in Dolly. Remember yesterday? I was going to say yesterday. No, last lesson we looked at Dolly the sheep, and so we need to just think about that same process of cloning, okay, but in plants. It's actually not the same process at all, okay. look, you can do one of two things. If you had a plant, okay, you can just cut off a little bit of the, take a small cutting that's going to grow, right, a little bit of the shoot, plant that in another bit of soil, and let it grow, and it will grow into a genetically identical plant, okay? But if you want to be precise about it, if you want to make lots of plants, lots of the same plant, okay, you can be fancy. If you had money, you can do micro propagation, okay? Micro means tiny, propagation means to make lots of. Okay, so what we're going to do here, we're working on a cellular level in a lab. So you would take some cells, shall we say, take a few cells from the plant, the snapdragon, yeah, okay, and then we would want to make sure that there is nothing on this plant that might interfere with its growth, okay. This is, by the way, I'm going to give you the most detailed answer possible here. So step one. Step two, we want to sterilize these cells, so maybe wash them in alcohol, or disinfectant or antiseptic. So sterilize these cells. Okay, you can just say sterilize, or you can say wash them with ethanol or disinfectant. Okay, now in a lab, what do we call that jelly stuff that has lots of minerals in it, and is like the perfect medium for cell growth? It is indeed agar. So place them onto agar jelly, because what does the agar have? The agar provides them, this provides them, what's the most important mineral, not mineral, sorry, nutrient that they need? Thank you. I'm thanking you guys for phantom answers. With glucose, or you could say amino acids. You can say minerals, but I would shy away from that. I would just state the obvious. Okay, glucose needed for respiration, amino acids needed for protein growth. So, done that, okay? And that's it. You just let them grow, and then they will grow into tiny little plantlets. And when the plantlets are sort of strong enough, you can take them out of your petri dish, your agar plate, and plant them into soil, and you'll have lots more of them than if you just took a cutting, okay? Because if you have one plant, realistically, how many cuttings, how many shoots can you cut off? Not that many. But with cells, obviously, it has hundreds, millions of cells, so you can actually do quite a few at once. There is also, just as a side point, is to remind ourselves of Unit 2. What's that growth hormone in plants called? The one that allows shoots to grow towards the light and roots or towards gravity. You might want to add that as well. So this is technically an extra mark here, but you can say add plant growth hormones, which we call, in this case, auxin. There's actually another one, gibberellins, but you haven't learned this and we don't need to know it. So auxin. Done. Okay, this is a very detailed answer. You only need 3 of those points to get the full marks there. Anyway, right, so now the next thing is scientists investigate the effect of exposing Snapdragon X plants. Oh, by the way, you know the tiny little plantlets that you get? You call them X plants, okay? The little cells that you take. These little samples that you take, those are called X plants, okay? Okay, so they exposed it to increasing amounts of ionizing radiation. Very random, not sure why we have radiation, but we got to work with it. So, take a snapdragon plant and use micropropagation to produce many X plants. Expose groups of X plants to different amounts of ionizing radiation. Okay, this is our independent variable. Grow the X plants into plants. Record the number of differences in their phenotypes compared to the original plant. So the number of differences in their phenotypes is my dependent variable. Take samples of each of the plants and measure the number of differences in DNA nucleotides. Okay. All right, so this is another one. There's 2 dependent variables here because we've got phenotypes and we've got DNA nucleotides, okay? So it says, give the reason why micropropagation is used to produce the plants to be tested. What did I say? Actually, no. I was going to say, what did I say propagation means, but let's state even more obvious answer. This is a type of cloning. Therefore, all the plants will be genetically identical, which makes is a control variable, right? We're keeping all the plants the same in the most same way they can be. All plants will be genetically identical. Okay, what term is given to all the DNA in an organism? I shouldn't even have to tell you this. Okay, it is genome. And now we've got a graph. We love a graph, don't we? This is an AO3 question. Straight away, I'm seeing the word discuss and a graph. So, amount of ionizing radiation as it increases. When we've got 2 things here, the number of changes in DNA and the number of different phenotypes. There's two things here. And to be honest, as you increase the amount of ionizing radiation, what happens to these two things? They both increase. So let's start with the obvious. Let's state the first thing that we can see, the pattern, the lines, yeah? As you increase, or maybe not as you, as amount of ionizing radiation, I'm writing as fast as I can, increases the number, I'm going to write NO for number, okay? The number of changes in DNA and, so we can say both, and number, because that is the overall pattern of different phenotypes increases. Okay, done. We've stated the obvious. Now let's think about the less obvious, right? Which one increases more from these two lines? So the solid line, right? Number of changes in DNA. So maybe there is a greater increase. There is a steeper, let's say steeper because the line is steeper, increase in changes in DNA compared to phenotypes, yeah? Now let's think why. This is unit 3, protein synthesis, okay, mutations, right? So ionizing radiation causes mutations, right? Okay, so let's state that obvious. Maybe it's on the marks, we'll check in a second. Ionizing radiation causes mutations, which are changes in the DNA base sequence, which then can change the what that is coded for? The amino acid, okay? So, which can change the amino acid that is coded for. However, clearly, if that is the case, if it causes a different amino acid and causes a different protein, then the phenotype would be different, right? But what this graph is showing us is actually there are a lot of changes in DNA in the base sequence, but actually not as many changes in the phenotypes as you would expect. So this is that question of why don't mutations always lead to a change in phenotype? Because it might not actually, it might not have an effect. It might just make the same amino acid, or it might make the same protein. Okay, so some of these mutations will have no effect because they may end up coding for the same amino acid or the same protein or enzyme, yeah? Done. 4 marks. Okay. Oh, another thing we can say is that, even if there is a mutation that changes the protein, if it's on an allele if it's in a heterozygous individual, okay, if it's on the recessive allele, then it will have no effect. Okay, so maybe the mutation is recessive. Let's just add that in as an extra thing. Mutation may be on the recessive allele. Okay, done, right? So that these are the paper 2 questions. Now, also with this lesson, I've uploaded paper 1, unit 5 questions. So all the combined stuff, the combined and triple, the stuff you can get on paper 1. I want you to use what you've done here, the way you've thought here. I want you to apply that now to the paper 1 questions for this same unit. There is a lot more AO1 there, so we're thinking about things like the lactobacillus bacteria, fermenters. Oh, I didn't see any questions on fermentation. Maybe I need to add those as well. Okay, fermenters, increasing crop yield, genetic modification. So try those, please, and then I will see you on Tuesday to go through those questions and any things you got stuck on. Thank you for listening to 45 minutes of me. The lesson is supposed to be 40 minutes, and that's what you get. You're welcome.